Ladies and gentlemen, good afternoon. I'm Mart Saarma. I'm working as a research director at the Institute of Biotechnology, University of Helsinki, and as Henri Huttunen said, it was our lab who originally discovered CDNF. In my short introduction, I will tell you what is Parkinson's disease, and I will also tell you why growth factors have been used to treat Parkinson's disease, and then I will show you some of the data of the clinical trials on two of the neurotrophic factors. Next slide, please. So as you all know, in Parkinson's disease, dopaminergic neurons located in the substantia nigra pars compacta and projecting to the caudate putamen start to degenerate and die. And what is probably very important is that at the moment when the first motor symptoms, slowness of movement, resting tremor, rigidity, etc. appear, about half of the dopamine neuron cell bodies and even maybe 60 to 70% of the fibers have been lost. So the treatment of the disease actually starts quite late, when already a massive degeneration and death of dopamine neurons has occurred. It should be also noted that in addition to the motor symptoms, patients also have a number of non-motor symptoms, among them constipation, loss of smell, depression, and in later stages, significant decrease of the cognition are among the most important. Next, please. If we draw a very simplified scheme, then at the moment of the diagnosis or at the moment when the treatment of the patient starts, we have about 50% of the dopamine neurons still alive. They make dopamine and they release dopamine in the caudate putamen. But eventually also these neurons will degenerate and die. And then we have maybe 10 to 15% of the neurons which are still alive, still able to make dopamine, but have lost the synaptic contact and have a different degree of axonal degeneration. And then we have maybe 5 to 10% of the cells which have even lost the ability to make dopamine, but they are still neurons. So these three groups of dopamine neurons are actually our target. We want to keep these neurons alive, and these two groups which have lost the axon or ability to make dopamine regenerate. So the goal of our drug development is to prevent dopamine neuron degeneration and death, regenerate them, and to increase the functional activity of these neurons. Unfortunately, all current therapies available do not offer that. They offer symptomatic relief and do not arrest or attenuate the progression of the disease. Next, please. So one very important question is why and how dopamine neurons degenerate and die. So dopamine neurons, like all of the neurons, are non-dividing post-mitotic cells, and according to the current understanding, there is very little, if at all, birth of new neurons, dopamine neurons from stem cells. These neurons have extensive network of synaptic contacts. Some dopamine neurons make up to 500,000 synaptic contacts, which mean that they are very demanding on energy and intracellular trafficking, and therefore very vulnerable. And they make massive amounts of dopamine, which requires strong protein synthesis, and dopamine, if it gets oxidized, may be very toxic. So what we know is that there are about 20 genes, mutations in which are the reason for the familial forms of Parkinson's disease, which form maybe 5 to 10%. But dopamine neurons which die in aging, in poisoning, And in Parkinson's disease, why they die, how they die is not very well understood. So we can conclude that this is a very interesting area of investigation to understand why and how dopamine neurons die. One should also take into account that brain is a very complex organ. It has 10 in the power of 11 neurons, which make 10 in the power of 14 synaptic contacts. Imagine, this is roughly 10 times more than we have stars in the Milky Way galaxy. Next. So in late 50s, Italian scientist Rita Levi-Montalcini described a new group of proteins from mouse and rat, which were able to keep neurons alive. And she called these new proteins neurotrophic factor. The very first neurotrophic factor she discovered, she named nerve growth factor, NGF. And on the picture on the right, you see the upper picture. This is a chicken embryo sensory ganglion. If you keep this ganglion for a week in the culture, all neurons die. But in the presence of NGF, they all stay alive, and the NGF induces a beautiful outgrowth of neurons. So on the next slide, you can see the picture of Rita Levi-Montalcini in the middle, and his graduate student Stanley Cohen. They got in 1986 the Nobel Prize in Physiology or Medicine for the discovery of growth factor. And I show here also Victor Hamburger, in whose laboratory most of this work was done in Washington University in St. Louis, because already in 30s, Victor Hamburger, doing beautiful work in developmental neuroscience, predicted the existence of these factors. And indeed, this became true. Next slide, please. So neurotrophic factors, which are very important for the dopamine neurons, belong to the GDNF family. And there are two particular factors which have very nice effects on dopamine neurons. These are GDNF and nurturing. These two factors, although they act also on many other neurons, including motor neurons, sensory neurons, sympathetic neurons, can in culture maintain dopamine neurons alive and also protect them from death induced by neurotoxin. On the next slide, you can see a principal cartoon how the growth factor, shown here in blue, binds to the core receptor, shown in pink. Then this binding triggers dimerization of the core receptors, and then they recruit a transmembrane receptor called RET, which after binding gets activated. And the activation means that in the cytoplasmic part of that receptors, tyrosine residues become phosphorylated, and these phosphotyrosines become the docking site and activation site for intracellular signaling. Two major pathways are activated by these receptors. These are MAP kinase pathway and AKT pathway, and these are the two crucial pathways which are responsible that neurons stay alive and that neuron axons regenerate. We know that quite well. Well, encouraged by these very beautiful data on the dopaminergic neuron cultures, scientists in Sweden and in the United States in mid-90s performed first animal experiments. On the next slide, GDNF or nurturing were injected into the striatum, the area where the dopaminergic neurons project. And then the animals were injected by the neurotoxin, which induces the death of dopamine neurons. And in these very nice experiments, several laboratories were able to show that in these moderate neurotoxin animal models of Parkinson's disease, both GDNF and nurturing not only protected, but also repaired dopaminergic neurons. Well, although these are very powerful factors, they have also limitations. They are very basic proteins. They diffuse purely in the brain. And then, according to the current knowledge, they do not work in all models of Parkinson's disease. For example, if you treat the animal model where alpha-synuclein, the component of the Lewy bodies, is overexpressed, then GDNF seems not to work. And also, if you induce a very severe lesion, so that more than 80% of the dopamine neurons are wiped away, these factors don't work anymore. So, next slide. Encouraged by these beautiful experiments in vitro and in animal models, several companies and groups conducted in the beginning phase 1 and then phase 2 clinical trials. We can now summarize that there have been 3 phase 2 clinical trials with GDNF protein and 2 phase 2 clinical trials with nurturing gene therapy. And to cut the long story short, we can see that in the 2 phase 2 clinical trials with the GDNF protein, no clear clinical benefit was obtained. Whereas in the last, which I will very briefly discuss, very promising results with GDNF were obtained. The 2 phase 2 clinical trials with a nurturing gene therapy gave statistically significant difference compared to the placebo, but the clinical therapeutic effect was very modest. Next slide, please. So in the first clinical phase 2 clinical trial with GDNF conducted by and sponsored by Amgen, GDNF was injected intraventricularly, and even at very high doses of the GDNF, no improvement in the motor score was obtained. However, at the higher dose, which was as high as 4 milligrams per patient, several adverse events were registered. Actually, we can now conclude that follow-up studies in monkeys have demonstrated that in this way of delivery, GDNF did not reach dopamine neurons. In the next series of experiments conducted by Dr. Steve Gill, Alan Hone, who is also today participating in our webinar, and the test sponsored by MedGenesis, intermittent GDNF delivery was used. In this case, once a month, patient received GDNF through 4 catheters implanted into the caudate putamen. And as you can see from the next slide, in this series of experiments, no side effects were obtained, and where all patients were pooled, the UPDRS score did not show significant difference between the groups. However, the post hoc analysis found 9 patients who received GDNF with a large, clinically important motor improvement. And what is probably the most important, in the positron emission tomography, in all of the patients who received GDNF, a significantly increased uptake of the fluorodopa was registered. So this is the first experiment where we see clearly positive signs. So what can we learn from these in vitro, in vivo, and clinical trials? Next slide. We can learn that actually in Parkinson's patients, the degeneration of the dopamine neurons and the loss of the fibers in the caudate putamen occurs quite quickly. I show here you the neuropathology published by Jeff Kordorev's lab, and you can see here in A, the staining of dopaminergic fibers in the control patients. Then next, 1 year, 3 years, 4 years, 5 years, and 10 years after the diagnosis. You can notice that already after 4 or 5 years, there are very little dopamine fibers in the caudate putamen, and at 11 years, the caudate putamen is essentially empty. So what does it tell us? Next slide, please. So it does tell us that that GDNF, at least in some patients, can slow down neurodegeneration. I think this might be the first time that the disease progression has slowed down in a chronic neurological disease. We have learned also that actually we should start the treatment as early as possible. Currently, because of the ethical considerations, it has not been possible. We should develop mammalian cell-produced growth factors and modified growth factors which have improved properties. Look at insulin, which is on the market. Using genetic engineering, insulin properties have significantly improved. We can use this for GDNF and nurturing. We can also develop compounds that can increase the levels of endogenous brain neurotrophic factors. Or we can develop small molecules that mimic the action of neurotrophic factors and do not require the brain delivery of the drug, but can be delivered systemically. Or then search for new neurotrophic factors. We have actually taken very seriously the last path, and on the next slide, you can see the schematic the drawing of the structure of CDNF, what Päivi Lindholm, at that time the graduate student in the lab, and myself discovered. And we also solved the structure of that molecule. And then we showed that this molecule has rather unique structure and also mode of action, and forms a novel group of unconventional neurotrophic factors which act on neurons completely differently compared to GDNF or nurturing. And one of the maybe most exciting property of this factor is that it does not touch naive healthy cells. So next slide. On the next slide, based on these encouraging results on animal models, including animal models of on non-human primates, together with colleagues, many years ago, we started a new biotech company, which currently has the name Herantis Pharma, and Herantis Pharma has started, among other things, to develop CDNF-based drugs. Thank you very much for your attention.